Here's a really simple and practical guide to how to design a screen. Now remember what you need to start off with in the first place if you're going to develop drugs is you need to start off with a target. And just for argument's sake, I'm going to assume that this is some sort of protein involved in signaling. Right? So what you're looking for is you've got a cell, you've got some sort of signaling protein, let's say it's a receptor, and you're looking for a chemical that will actually interact with that target and either produce a response or to block a response to something else. Let's assume that you're looking for something to produce a response is the easiest one. Now remember, you want to be able to screen thousands if not millions of compounds. So it's no good putting cells into test tube, adding drug one after the other and then looking for a response. You need to be able to automate the system. And the best way to do that is to have some sort of clear readout. In fact, you have to have a readout of some kind. So usually what you might do is you might say, okay, I've got my target protein, I'm going to transfect a cell using some sort of DNA technology, try and make that look a little bit more like DNA technology, so that now you've got a transcript and that this cell doesn't usually produce the target protein, so you've now got the cell targeting the protein, and you might want to link this to some sort of fluorescent signal. So maybe what you do is you might maybe engineer this protein so that when it is activated it produces some sort of fluorescent signal. So then, if it should so happen that you find a compound which does indeed activate the receptor, you will then see some sort of fluorescent signal. And then what you can do is you can then automate the whole process by putting your cells into plates with thousands and thousands of wells. You put your cells in there, in each one, and then you automatically add chemical after chemical after chemical using robotics. And then hopefully you will find, so this has got thousands of wells, you'll then find one of them has what we call a signal. We might find one here, another one there. It's usually one out of getting to the order of tens of thousands of chemicals actually produce what we call a hit, which is when you've actually added a chemical and you end up with some sort of signal, some sort of fluorescent signal, because you've managed to find the 1 in 1,000 compounds that is actually hitting this receptor and causing this fluorescent signal. Most of the chemicals don't do anything. So for most of these wells, you see nothing at all. But on the rare chemical that actually works, you get a signal. And this is exactly what you're looking for. You're looking for your hit, as we call it. And that produces your lead compound. So, there are many, many ways you can create a screen. Right? So the most obvious one is to have it so that when the drug, sorry, not the drug, let's call it what it is at the moment, when the chemical activates a receptor, you get some sort of response, and that has maybe a fluorescent readout, or maybe it might bind to another chemical to produce a colour. So long as you can automate the readout, they can do thousands and thousands of drugs. That's the usual way of doing it. So for example, what you could do is you could have a cell which is transfected with your receptor, if this is a G protein coupled receptor. Let's say if this is coupled to GQ, you would then get a calcium signal. So then all you have to do is to put in a calcium sensitive dye which fluoresces in the presence of calcium, and then you go and do the same thing again. You have your plate with many wells. Put these cells in each of these wells and add your chemicals, one to each one, until you see one of them gets a fluorescent signal. So your imagination is all that limits how you can create one of these screens. There are other ones. So for example, if you were looking at cells from the heart, you can extract myocytes. These are the individual heart cells. And you might then use some sort of video recording technology and simply use machine visual processing to look for changes in the way they beat, for example. So long as you can automate it. Secondly, so as long as you can add thousands if not millions of chemicals. And thirdly, you want to be able to see an effect. Now what do I mean by that? You need to have a readout which is very clear so that there's a big difference between the wells which have no response and the wells which have a response. So in other words, you want to have a big gap between whatever your readout is for a no hit and a real hit. So that's how you design a screen.